discussion and a question-answer session. So thank you very much.
Well, thank you all. Thank you, Ali and Lisette, for the invitation. It is an honor for me to participate in this seminar and to change information with you. And so I will tell you next when to change. Next. Then I wanted to tell you that nowadays I'm living not in the capital city. I am in Rocha, as you said, Ali, and I am working in the media department. It is a department which the focus is data modeling and using statistical and artificial intelligence to analyze these different processes, including community, biodiversity, and water quality, among others. And to share with you some things, I was thinking how to start, and I want to start with the PEG model, with some pieces of the first publication of the PEG model. Then I will go through the development of the morphology-based functional groups and some applications, and then finish with some thoughts about how to contribute to these problems we are now facing in our area. So please, next. So the questions we want to investigate and search for responses are the same now than in the early years, as Reynolds said in 1988. So we are still going in the same path, trying to describe what lives where, why and how those organisms are living where in these communities, focusing on the mechanisms, and also to, and nowadays is more and more important to be able to predict what is going to happen with these communities in view of the great changes we are facing now in the world. So we were working with different researchers from the area for a long time, trying to organize the information to answer these questions that we all, I think we all are still looking for the answers. Next. So I wanted to start with the plankton ecology group. For me, it was part of the basis of my formation as an academic. I read many times this paper and others related, and then the papers that arrived afterwards. And I still find in these original papers a lot of information and ways of doing science that are really important and still contribute to future researchers and actual researchers. So this PEG model was constructed in these type of sessions within the SEAL with researchers that try to summarize and simplify the multiple dimensions that determine how phytoplankton communities or plankton communities or freshwater communities organize in different lakes. So I think we are still in this way trying to simplify, but considering more and more complex interactions, but still working with this type of approach. So I wanted to start here. Next, please. We are very, very familiar with this type of presentation of the information. It's very clear. It gives us a lot of tools to start with and to evaluate, generate hypotheses. And in this model and others related to this first work, the information is organized in different steps, including what we then call traits, functional traits, like small size or large size, fast or slow-growing species, organisms, the presence of resting stage, the different filtration rates, or the capability of eating or not different algal species. These are all traits, morphological or physiological traits that helped us, and in the past and in the future will help us to summarize information with focus in significant and relevant mechanisms. Next, please. So what the authors said at the last part of the manuscript, the paper, was that future authors are invited to adopt this approach in which all the major pathways of plankton succession are examined by means of statements of this occur when this will happen. So it's an approach easy to apply and easy to use as a basis for analyzing and giving alternatives to understand and predict also what is going to happen in one lake in different situations. This model has been revised, expanded. Other communities, members of the freshwater habitat have been included, and nowadays is I think we can still contribute to work in this manner and in this approach. Next, please. So, what comes to my mind or to this presentation next? Other approaches that have been done or have been we have been working in, or different researchers have been working in, to still also summarize information and understand and try to predict. These are the phytosociological approaches that have a common origin in terrestrial ecology and have developed from animals and plants towards freshwater and marine environments, with the first contributions of McCartan and Wilson, Bianca, that identified the R and K strategies, and then the life strategies of Green, Sears, Air. And these were taken and adapted to other communities in the aquatic environments. Next, please. Margalef was a pioneer in this matter. He translated this type of approximation to both marine and freshwater habitats, and his paper on life forms of phytoplankton and survival strategies is still a source of information and a source of new ways of understanding and working with the assembly of communities. This presentation of the axes summarize and explains this temporal succession and contributes to understand when and why the harmful algal blooms are going to occur. So it's still a tool that we can and we have to take in account. Next, please. Then we have Reynolds, of course, with a lot of contributions that he did, particularly in freshwater habitat, but also in his late years in marine environments, when he proposed the associations that they were afterwards called functional groups. Next, please. Working with researchers from different regions, from Europe, from America Latina, and here we have Vera Husar, a fundamental researcher that in many ways introduced Latin American environments in this global phytosociological approach. So I think Vera is also here. And Isaac that also worked a lot in summarizing and organizing these approximations. Next. In more recent years, the ocean again was in the picture. In the beginning, the approaches were more united, considering both freshwater and marine environments. Then I think there were two ways, parallel ways of doing or developing these approaches. But in the last years, both environments again are being analyzed together, and this has is very important because the different ways in which oceanographers and limnologists view and understand and investigate how communities are assembled are complementary and are important to advance in the research. So I think now the picture is more complex, but also is more rich and has more tools that will help us respond these three groups of questions of describing, understanding, and predicting. Next, please. So just to summarize some of the concepts that are common to all these approaches, and I think is conceptually more clear or organizing in what we call trait-based ecology or functional ecology and functional traits. What we call functional traits, for example, size, growth rate, are physiological, morphological, and behavioral traits that originally are measured at the level of the organisms and have the good capability of representing fitness or explaining how the particular organisms are going to contribute to the next generation, how they are going to survive growth and performance in different conditions. So using functional traits or traits that describe fitness, we can take from these all approaches and summarize and reorganize information. Next. Then the functional groups are groups of organisms that have similar functional traits and that have similar fitness. I mean, they performance similarly in different environmental conditions, and also reflect the responses of the organisms to the changes in the environment and the effects of these organisms in the environment. And what this approach looks for is to quantify how these groups of these traits change with how much, which are the growth rates, which are the abundances that these traits and these groups are going to have with different environmental conditions. Therefore, we can feed this understanding and prediction with quantifications. Next, please. It is. Are you listening okay?
Yeah, I think there's a microphone open somewhere. If we could all turn off our microphones.
Yeah, I've been muting microphones here and there whilst you are speaking, so continue.
Great. Okay, so what these approaches have in common, the PEG modeling approaches, the phytosociological approaches, and the trait-based and functional ecology approaches, is that they focus on processes and reorganize information, key information, to describe, explain, quantify, and predict changes of communities in particular or diverse environments. Next, please. Within functional traits, morphological traits are like particular in that they are easy to measure, and they represent well, indirectly but well, fitness and performance. This is an image from Finkel that also shows that morphological traits represent the wide variability of condition and of the diversity of organisms that are in the ocean or in limnic environments. So also the great variability of morphological trait is an advantage of using them to evaluate community assembly and construct functional groups. Next, please. So now I want to share with you a past history of how we worked and constructed these morphology-based functional groups with the aim of summarizing, understanding and predicting how phytoplankton communities assembly in different conditions. Our hypotheses were that the responses and effects of phytoplankton organisms can be summarized using exclusively morphology. So we identify easy to measure morphological traits and measure them in many lakes in different latitudes, and then use different approaches, statistical approaches, to construct the groups. And the good thing of this is that we don't need the species identification, that you can measure them in a light common microscope, or nowadays you can use different cytometric or equipment to measure automatically also the organisms and classify them into these morphology-based functional groups. Next, please. So here are the 7 groups. Maybe you already know them, or maybe not. So I just am going to present them. This is group 1 to 7. Group 1, they are small, rapid growing, high SV surface volume ratio organisms. Group 2 are small silicious flagellates. Group 3 are large, high SV, high surface volume ratio filaments. Group 4 are medium size with no categorical specializations, categorical traits. Group 5 are flagellates of medium to large size. Group 6 are non-flagellates with silicious excellent. They are mainly diatoms. And group 7 are large mucilaginous, low sulfate volume ratio colonies. So these are the 7 groups we constructed. Next, we are going to show some results about these groups with focus on group 3 and 7, because these 2 groups include cyanobacteria, and cyanobacteria that can form nuisance harmful algal blooms that are causing a lot of problems along the world and in our region. So the examples are going to be analyzing, taking in account these 2 groups. Next, please. So what is this classification about? You can easily classify your organisms in these 7 groups, and these 7 groups have different physiological rates, like maximum growth rate, like sinking rates, and they are associated to different ecological performance. So, for example, group 3 occurs in has a medium growth rate, low sinking rates, or can select where to be in the water column. And group 7 are these large mucilaginous colonies that have very low growth rate and, for example, high saturation concentrations of resources. So different groups have different functional groups, functional traits, and as a result, different physiological responses and also effects. Next, please. Here are some examples. These are the 7 groups and the differences in growth rate. Next, please. Or also here the differences in the light saturation constants. And you can see group 3 that is typical of turbid ecosystems, while group 7 can form surface blooms and perform well, better with high light conditions. Next, please. Then these groups, due to the differences in morphology and also the other functional traits, they have different sensibility to zooplankton grazing rates, to zooplankton grazing. So different communities of zooplankton can affect more or less these organisms. Here also number 3 and 7. 3 is easily or more eaten by copperbots, while 7 is not eaten at all by copperbots, for example. Next, please. Also, what was very interesting was to see that also these 2 groups, these are the 7 groups, and the growth rate with temperature. You can see there in the x-axis the temperature and in the y-axis the growth rate. Different groups respond differently to the increases in temperature. For example, group 6, that are mainly diatoms, they increase their temperature until 25, and then they decrease, while group 3 and 7 increase more than the growth rate above 25, but as we can see there, they respond differently. They are both cyanobacteria, but group 3 increase the temperature until 28 and then decrease, and group 7 can still increase the growth rate beyond 30 degrees. So this also contributes, for example, to analyze what is going to happen in lakes where the temperature are still increasing. And you can see that not any cyanobacteria will be dominant, but different ones here represented in this different classification based in morphology are going to respond to these temperature increases. Next, please. Then we analyze how they change in particular environmental conditions, and here again we have a card. It's a classification and regression tree. It's a machine learning model, and we analyze how these groups were dominant in different conditions. And here again you can see you can follow the path and identify which were the environmental values that presented different thresholds. So different groups occurred in different conditions. Next. And also different groups occur in different stages of the succession. Next, please. Then we also looked at how these functional groups, morphology-based functional groups, change across climates, and we find that in all climates, these were lakes in different areas of South America, and we found that the groups were present in all climates, but the species richness changes among climates. And when we investigate within each group, next please, we can see that different groups respond differently. The species within groups respond differently to different climates, increasing or decreasing the internal richness. So you have all groups in all climates, but within the groups we also have variability in the number of species. Next, please. Then we started to analyze estimate the fitness, because we are talking of functional group, functional traits that have to be related with fitness, and we wanted to estimate fitness. So with different equations, and based on the main mechanisms that define how phytoplankton can survive, growth, and perform, we estimated the growth rates and the responses to different conditions, and estimated the R-Tillman as an indicator of fitness. Next, please. And this allowed us to predict along wide environmental conditions which groups was going to be dominant. For example, here, this is a model for a shallow lake where we change the flushing rate in the x-axis and the grazing pressure in the y-axis, and we can identify in which conditions are we going to find different groups. In this lake that we were trying to manage the lake to improve the water quality conditions, we, for example, tried to change the flushing rate to move the community towards the dominance of group 4 that has no problems for water quality. Next. Also, we investigated how temperature can affect different groups' performance, and we found that a higher temperature, particularly group 3, in conditions of low light, this is just an example, it was going to be dominant, but when temperatures were high and light was high, group 7 was going to be dominant. Next. Well, with all these tools, we then moved forward to investigate how community assembly. This is a kind of similar approach to the one that the PEG model applies, that is a series of steps that when this occurs, then this will happen. This is a phrase from the manuscript of the paper of Sommer et al. So we use this context, these tools, and these investigations to generate a process or a series of hypotheses of how community of phytoplankton ensembles in a particular ecosystem. So next, please. First of all, you have a species pool of a species in your region, where all the morphology-based functional groups are represented, as we show with different climates, and local conditions are going to determine which one of the morphology-based functional groups is going to be dominant. So we postulate that the classification into these groups also allows us to determine the niche in which the species or the organisms are going to be dominant. So, in particular environmental conditions, one of the 7 groups is going to be dominant, or is going to be able to grow. So this is the first step. Then the next one. Next, within groups, we suppose that most organisms are equivalent. I mean, any of the organisms with the traits that are described in the groups is going to be successful in this particular environment. So we postulate that there is a functional equivalence within the groups. This is the second step. So first, based on the local conditions, one group is selected. Then within the group, any of the species or the organisms within the group is going to be successful. Next. Then you have a community that is assembled You have a dominant group, and you will have different invasions. So, which invasions will be successful? The first hypothesis is that the species that will be successful will be that belongs to the local, already dominant functional groups. The second one is that other species that are not from the same group or belong to the same group and have particular combination of traits will be dominant over the resident species. And the third is that if you are receiving the species of any group with a very large inocula, you will have this species as dominant independently of the local resident morphology-based functional group. And finally, next, we are considering and taking into account that local already established species might cooperate with invading species and therefore somehow contribute to their development, successful in the development in the ecosystem. So next, please. So we did some experiments to evaluate these steps in the community assembly. The first one were experiments where we constructed the different environmental scenarios with or without nutrients and low temperature, without nutrients and high temperature, with nutrients and low temperature, and with nutrients and high temperature. In the number C, figure in the panel, represent all the figures represent the final state after between 10 and 20 days at the end of the experiment, because we constructed a diverse pool of species and inoculated them in many flasks representing these 4 different conditions, let the time go and analyze it along the succession and at the end of the succession. What we found is that, of course, at high nutrients, we have higher biovolume, that at a low nutrient, and also that at high nutrient and depending on the temperature, we had different dominating functional groups. At 14 degrees Celsius, we had group 6 dominated by diatoms, and at the same nutrient concentration, but at higher temperatures, 26, we had group 3 dominating. So we were able to represent this first step of the community assembly with different environmental conditions. Next. Then we wanted to analyze if at the end, this is in the case of the high nutrient, high temperature conditions. Next. Yes, this one. Here in the first graph, you have the replicates of the treatment with high temperature and nutrients. So there are 4 replicates, and in all cases you have group 3 dominating. The explanation of group 3 domination is the high temperature, high nutrient, and also the low light within the experiment due to self-shading, that only this group with this morphology is capable of develop. Then we looked at how many species were coexisting in each replicate, and we had from 2 to 5 species when we looked at the microscope, but we also analyzed this with molecular approaches, and we found like 31 species of the same functional group number 3. So many species were coexisting within each flask. Then we looked at which were the species, in each flask that were dominating, and we found that different species from the same group occurred in different replicates. So somehow we are supporting the hypothesis that within morphology-based functional groups, species are functionally equivalent. Next. Then we try to see what happened when you already have your community assembled and you made some invasions. So we did this 3 treatment. These represent the temporal changes in total biomass. In 3 treatments, the first one where we didn't do any invasion, the second that we did an invasion with a species from the local dominant functional groups, and then an invasion with a cyanobacteria, but not of the local dominant group. Next. And we found that, as expected, the successful invasion was that of the species of the local dominant group. Please, next. This is a lot of information. And then we move to, and this is what we are actually working with our group, also with Melina and Claudia and Vera, and the group of the Microsudaqua. We are trying to discuss and work on this paradigm of competition versus cooperation. We want to investigate and take into the discussion the relevance of cooperation for phytoplankton, for microorganisms in general. Next, please. So what we did was to analyze these different treatments with different invasions in terms of the functions that were represented in these different treatments. And we found that found that different treatment had similar functions. So despite the composition was, in terms of a species, was not always the same, we did have always similar functions. So this also contributes to this functional neutrality or the maintenance of fundamental functions within the same functional group. Next, please. Also, we are trying to identify with molecular analysis if there are traits that show the cooperative interactions. So we are looking at which are the traits and the functions we see in the genes that can be associated with cooperation among organisms. Like, for example, nitrogen fixation, the assembly of siderov for interchanging ions or other functions that can be related with cooperation. Next, please. We also analyzed if there were species that were particularly different from these groups, because until now we saw that local conditions define which group is going to be dominant, but once you have one group, species are functionally equivalent, but also there might be some species that are particularly different to the rest, and therefore they are called as invasive species. So we're investigating Serratium fulcoides. That is one of the species that is actually invading. Sorry, my cat is crazy. Sorry. So we're investigating this particular species that has a particular characteristics. Next. Here, for example, we can see, we compare this Ceratin furcoide that is part of morphology group 5, but there you can see in the histograms, in the density plots, that in red it is Ceratium, and in black is the rest of the flagellates in this group. And Ceratium has always particularly larger and characteristics that make it particularly different to the rest of the organisms in the group. Next. And we estimated the Tillman R and compared, we estimated the Tillman R for different legs and condition in the legs, and we saw that it has higher fitness, and when it has higher fitness, it has it has lower R, higher fitness, and higher abundance when compared with other species of the same group. And for example, one of the pressures that don't limit these Ceratin furcoides is grazing pressure. In this type of legs, grazing pressure limit other species of group 5, but Ceratin furcoides, due to the high size, escape the pressure and can become a dominant species and also invade in this type of ecosystems. Next. Well, now getting to the last part of this presentation, we here in my country, are also in the world, but also particularly in South America, we are facing a lot of very important water quality problems. This is an image of a harmful cyanobacteria bloom that attained more than 800 kilometers along all the coast of Uruguay. Next. So one of the things we are doing, and we are focusing more now, is in applying these approaches of trait-based approaches to monitoring, predicting and managing these harmful algal blooms. Why? Because we can synthetically summarize information. They are groups easy to recognize also by persons, technicians that are working in the institutions monitoring these problems, because we can list the preferences and the sensitivities, and we have and they have similar effects, similar responses to environmental changes and similar effects, and it helps to reorganize the work in different stages. Next. Here we have the list of the seven groups, and we have been working, this is in Spanish, sorry, describing these groups and identifying the effects of these potential effects on these groups, and also which measures we can apply to prevent the negative effects on different uses of the water from the ecosystems. Next. So what we have been doing also is to integrate different levels of functional traits. We have been working with cells, individual colonies, on aspects of physiology and morphological traits. Then we are working with the populations, with demographic traits, and trying to next to integrate this information towards the community, as we have seen until now, but also at the ecosystem, to evaluate, predict the development of toxic surface blooms, for example. Next. So we can integrate this information in the cells and in the individual traits towards the prediction of these massive blooms in large ecosystems. So this is one of the aspects next we are working in. Also, trying to move, the previous was talking about going from the population, our organisms, to other ecosystems. Also, we are working within the groups. For example, this is group 7 that forms Microcystis aeruginosa complex is a very common, global, commonly found harmful forming of harmful blooms. We are investigating how different sizes within this group have different effects. So, for example, here we have in the graph up, we have different sizes that are represent different steps in the life cycle of the complex, and besides below how these different fractions of size fraction have different production of ARN for the production of microcystinas. So we can measure sizes and have a diagnosis of how actively these organisms are producing toxins. Next. Also, using morphology, morphology explains, represent well toxicity, and morphology can be measured with remote sensing. So, because different morphologies have result in different optical properties. So, we have been working with this. Oops. And we found, next, and we found that different morphologies reflect different toxicity, and different morphology reflect well different optical properties, and developed an application for a cellular form to make for citizens measure the probability of having toxic cyanotoxic in the water. Next. Next, oops. Well, then, as we are working in different levels, we apply this also to monitoring and alert systems, to look first at the ecosystem, then take samples with nets, then look at the microscope, and then analyze by qPCR, and also combine these different levels of monitoring with different levels of alert, and integrate this information to help taking decisions in, for example, recreation waters or during potabilization of water. Next. So we have been working a lot with problems of health. First, since 2000, we have been increasing having cyanobacterial blooms with clear effects on health. And 2 years ago, we had problems with quantity of water, and we didn't have potable water for 6 months in half of our country. So these are problems, very serious problems that we are having, that we have to work and design tools to try to contribute to these problems in our countries. Next. So how are we nowadays working? We're trying to continue with this functional ecology approach, with campaigns in the field, with experiments, with models, but also next, taking in account what is going on in the catchment areas, how the changes in the use of the land is affecting our water quality, and next also including studies with the people that is affected by these water quality problems, and that know how to recognize the problems and when they occur. So we are trying to combine all these perspectives and tools to look for solutions or contribute for solutions. Next. Well, I wanted also to take here to the seal, because it's an international space where it would be very nice if we can contribute from here, because we have a problem, a great problem now, that is many countries in the world, especially in in Europe are improving water quality, but many countries in other areas are not improving, but letting worse in the water quality. It's just a review of how the percentage of oligotrophic, mesotrophic, and eutrophic lakes change in different continents. Next. And here I show you two examples. This is how in lakes in Swiss, but there are many other publications are improving water quality, especially since 2000, where the lakes are going from eutrophication to re-oligotrophication. Nevertheless, in our area, this is at the right study we did in Uruguay that has a catchment area that includes Uruguay, Argentina, Brazil, Bolivia. And as you can see, since 2000, our water quality is getting worse and worse. So we had these two sides of the same coin. I don't know if it says the same in English, but this is how I translated from Spanish, that in some places of the world, the water quality is improving, but in other places of the world, the water quality is getting worse and worse. Next. One of the explanations is that we are using more and more our land to produce crops with a lot of fertilizers, while in Europe, for example, the land use and the percentage of fertilizers are diminishing. Next, please. And despite this, that we could think, well, we have more fertilizers, we are producing more, we have less poverty. No, we are more and more poor, and our countries are having less water quality, more health problems related to water quality, and are more poor. So what can we do about that in the seal? Next. So some questions here I take for you to consider if you want to work about them. One, if it is feasible to work with trait-based approaches across aquatic communities, considering different tools, organization levels, from genes to the ecosystems, and also considering the impact of human activity on these communities. Can also contribute to our understanding of community assembly and ecosystems responses, and identify potential alternatives to improve water quality. Can we contribute from the PEG group in the seal to use these approaches to answer relevant questions to actual problems in our countries? Which is the state of the art of the trait-based approaches in this sense? Can we adapt PEG model to include anthropogenic problems? Next. Also, I wanted to share with you that we were interchanging with Simone and Marie about this use of traits to discuss some of these questions, and maybe we can make a group within the PEG to advance in these aspects. Next. Thank you very much, and sorry if it was too long. Thank you.
Thank you very much, Carla. That was great. I mean, we just had an amazing lesson, basically, on phytoplankton functional traits, its evolution, theory, application, all the way to global justice issues. So that was really nice. I think we still have some time for questions, and the ones you raise, I fully support. Carla, we can talk about it later too, but we can discuss it here as well, how we can include and join forces with the PEG and dive deeper into some of these issues. So now the floor is open for questions. You can either ask opening your microphone, or if you don't want that or you can't for some reason, then the chat also is fine. We're receiving a lot of praises in the chat.
We have one question from Simone Cardoso.
Hi, everyone. Thanks for being here and watching Carla's presentation. Carla, it was an awesome talk, and I'm just thinking about all the subjects that you brought and the new methods that we have to study plankton. And I would like to ask you if we need more understanding, we need new methods to understand plankton, or we can answer our questions with the methods that we already have. And I would like to hear you talking about it, and how do you see these things? Because sometimes I see people working with microbes and they are using like very fancy new methodologies, and do we need it? Do you think it's the time to incorporate this? And thanks for your nice talk again.
Thank you, Simone. Nice question. I think all new methods are welcome. First of all, that is important. But also, I think we can answer most of the questions with the methods we already have, or with simple methods. Not necessarily we need very complex and expensive methodologies. So I don't know, what do you think about it? I think that we need to work with what we have already. That is a lot. And also, if you have more information, it is good to incorporate it. But maybe it is difficult for some groups to incorporate these new methods because they are very expensive. But nonetheless, these groups can work and generate new answers and can still contribute with equal or more good investigations. Also, it happened in our countries that we cannot afford some of these techniques, but that doesn't mean that you cannot work with any question. And that could be very, very bad that you cannot publish, for example, if you don't use that kind of method that you cannot pay for. So this is another discussion, to publish or to perish, or to publish where and why. Also, we are publishing in English, that is not our language, in magazines that are not read by our people because they don't understand it. I mean, you have to see, try to do what's best as with different tools you can have. Sorry, this is the more or less the idea.
So the next question is in the chat from Michael. He says he thanks you, and then he asks, what would be the minimum equipment needed to start working this way?
The minimum equipment? Your computer. And a paper and a pen and a group, also a group of, not alone, you need a group to talk, to discuss, to open your mind. A paper and a computer, this is the minimum equipment, I think.
The next question is from Cielio. He also thanks you for the great talk and then asks, what's your opinion and the progress on assembly rules of phytoplankton, specifically Reynolds functional group and morphology-based functional group, using null models? Are they always needed, or can we estimate limiting similarity or niche filtering without them?
I think you can do both. Yeah, using null models is one approach. There are many approaches to investigate this. Also the same, you can try it with different approaches. Using new models is one, but it's not the only one. If anybody else want to comment, please, you're welcome.
So Jens just commented that with the growing artificial intelligence, maybe some of the more expensive methods will be cheaper. And Gülce has a question. What do you think about the trade-off between ecological simplicity that the morphology-based functional groups framework and the species level nuance, especially when dealing with multifactorial stressors?
Yes, the idea is not to use only the morphology-based functional groups. Just the morphology-based functional group is like one step, and then you can subdivide, or you can analyze traits individually, or you can use other approximations. The idea is not to get stuck with something, but to try to use a combination of things. This is one approach that you can use, and then, for example, within one group, you can investigate differences with different stressors. This is just a simple way to maybe to start.
Okay, thanks. And Ulrike, I think she raised her hand.
Impossible to unmute.
Now it works. Okay, yay.
We hear
Now
you.
it works. Now it works. Okay. I have a comment. I'm working more with zooplankton, and when you're talking about rotifers, then I can add that also rotifers have preferences for feeding, and I guess everybody uses Brachionus for their experiments, but Brachionus is only one genus, and probably it's Brachyolus caliciflorus, so it's one species of over 2,000 species. So my comment is that it's time to combine zooplankton functional diversity with phytoplankton functional diversity. And it's also time that people that work with algae also think about other rotifers than Brachionus, other cladocerians than just Daphnia. And that's a comment. I mean, it's a comment, an invitation that people that work with phytoplankton collaborate more with people that work with zooplankton, and that they try to make a model that looks at both aspects. So how do the traits, the phytoplankton traits, influence zooplankton traits, and vice versa, because they all depend on each other. This is just a comment, and it's just an invitation to be more, to have more active collaboration between both sides.
Great. I just wanted to want to tell you that we also work with natural communities. Not only the experiment I show you were done with natural communities, not with cultures. So we went to 20 lakes. From the lakes, we made the inocula, and we took the zooplankton, all the zooplankton that was there for the experiments. Also, the other work was done with with many species, not only with a revision of different species. It is true that there are more works with some species, but yes, the idea, I think it would be very nice, is to work with the communities, with the associations that you found in the communities. So plankton with phytoplankton, also with bacteria and virus that they come with them, and you don't know. And also we are also working with the bacteria that live in the mucilage of the microcystis and how they change with different sizes. So yes, this is a it's a diverse approach. We have to combine all. Thank you.
That's a great comment, Ulrike. Trait combinations. I mean, the PEG is the perfect place to bring up these ideas because we can actually get together and start working on these. So just a reminder for everybody, if you're not already joining on our online ideas list, you can put these ideas and we can actually help move them forward. But anyways, our time is kind of drying up, but we have still some hands. So I think Stella or Jens first. I forgot.
Yeah, so me and Jens on Stella's. So I like Enrique's comment because we are talking about traits here, and we forget the ciliates and like larger copepods eat the smaller ones. I think we should think about more trait groups of predator in this because it's a long row. And maybe we can't look at all the species, but we need to look at, it's not just one zooplankton eating one phytoplankton. Especially for us from marine sites, we know that there are many sort of, you're balancing on several balls here, and depending on where the peak, it sort of focus on. So we need to look at several steps of predators because they eat each other, and it's sometimes very important, like a ciliate and a large copepod in marine system has completely different effects. So that's one thing. And the positive thing, as I wrote before, the imaging, even in situ, is getting cheaper and much better. And all the programming gets lighter. We can do it on normal machines. So that is something that soon should be possible to do all over the world. And the good thing with image data is that they can be taken in one place and can be analyzed anywhere else on Earth, and they don't deteriorate like samples do. Or if somebody counted the sample, we don't know what was in there, but if you have an image, we can look at it 100 years later. So I think that way is something we should try to go. And there are even cheap machines like, Planktoscope and others, not the best, but they cost a few thousand euros. So, and this is moving fast. So I think we should look at imaging. There's much more quantitative than molecular approaches. And for the larger organisms here, at least, it's definitely a good thing to do. Yeah, just that. Thank you.
Okay, so back to chat now. Gülce has a follow-up. In salinizing freshwater systems, which traits or functional groups do you expect to be most strongly selected besides size?
We are not now working in estuarine ecosystems and in the coastal ocean. I think size will still be a very important trait. Size form will still be an important trait. We will probably have a wider range of sizes also in salinized or marine systems, larger organisms, and wider by diversity in forms of, for example, diatoms. And also dinoflagellates are a complex group, especially in marine ecosystems, because you have other traits that maybe are not easy to identify, but we can go to and try to, for example, heterotrophy or phagotrophy or some things, and they are really bizarre, the dinoflagellates in the ocean, especially in the coastal ocean. But there are some works of Smida about dinoflagellates that we should go and relook again to continue with this approach.
Can I ask a follow-up question, if I may? And promise this is the last one.
Shoot ahead.
So, so with the new imaging techniques, we can have many images, but we can also have data in addition to the images, right? Is there any trait that in the past you were thinking that it's hard to measure morphologically, but now can be used with these data sheets? For example, we use FlowCam here. We can have more numerical values, especially for symmetry, for color. Would you like to include some of these in your sheet?
Yes, it would be great to include, for example, heterotrophy, autotrophy, color pigments, of course, especially for marine ecosystems. Yes. Activity also, I think you can have activity in the images, the activity in the DNA or something like that. Yes, yes. Anything, all is welcome. I think. I think heterotrophy, this is a very interesting thing that is not easy to identify under microscope.
Thank you very much.
Parasites also, they are saying there. Also interaction with bacteria, like this, I don't remember the name, this layer it forms around the organisms that is a site where they interact with them and also with the media. Will be interesting to measure it also. Maybe you can measure this area around each organism.
It's really nice to see. It's really nice to see this great discussion, and I'm sure Carla would be happy to answer these questions offline. But I think the last question is going to be Dorte, who raised her hand, just in question of time.
Yeah, thank you very much. Your morphology-based groups are very different, where some have only cyanobacteria in it, whereas others have a full range of systematic groups. What do you think is the impact of systematic group being in one of the morphology groups, whereas others are very diverse? So what's the impact of the systematic affiliation to the group versus the morphology?
I think it is independent. The thing is that different phylogenetic groups have more or are more or less diverse. For example, cyanobacteria are very diverse. You have from very, very small oligotrophic species to very, very large bloom-forming ecosystem change, altering the whole ecosystem in one phylogenetic group or taxonomic group like cyanobacteria. Then in freshwater, dinoflagellates are less diverse, but maybe if you go to the marine environment, you will have different species of the same phylogenetic group in different groups. Also within cyanobacteria, Microcystis aeruginosa can be in different groups because you can have it like individual cells, or you can have it in medium-sized colonies, or you can have mega big colonies. So this is good that there is not an effect. It just represents what happened within phylogenetic groups. I don't know.
But on the other hand, if you have, for example, the ones with the trichomes, which consists mainly of cyanobacteria, or other ones which mainly consists of diatoms. So I mean it the other way around.
I don't understand. I think diatoms, what we did was to construct a matrix with all the size measures and categorical traits presence, and statistically separated them in these 7 groups. And with this type of organization, you can represent in a coarse manner what is going on in a lake or whatever. It doesn't affect, I think, the groups. But maybe I don't understand your question.
For example, when you were showing that the one group which were mainly dominated by diatoms had these decreasing, or they decreased with temperature increased. And then my question is it a matter of the size, so of the morphology-based functional group, or is it a matter of that diatoms as a systematic group cannot hold this high temperatures?
Yes, it goes both. It goes. You cannot separate them. It doesn't mean that it is wrong to separate them by size. Just that the morphology is just that diatoms as a morphology group and a phylogenetic group are homogeneous. They have similar behavior if you classify as diatom or classify as with the morphological group. But cyanobacteria, if you take them as a group or you separate them in many functional groups, is different. For diatoms, it's the same. For cyanobacteria, it's different. So you get independent from phylogenetic groups when you organize organisms based on functional morphological traits.
Thank you.
You're welcome. Good question.
I feel terrible to say that we have to wrap it up because the discussion is really going, and that's what exactly this is for. So I'm sorry from everybody whose questions we're not going to be able to include now. But let me please remind everybody again to continue refining these ideas on the online document that I posted in the chat, if anybody has ideas, and also to suggest future presenters for this webinar series. We're totally open to people's presentations, suggestions from you guys. So we want this to be a community-driven, bottom-up process. So thank you very much, Carla, again, for joining us. Thank you, everybody, for participating. It was great, and I'm looking forward to the next one already, and also thinking about these ideas.
Thank you very much.
Thank you.
Thanks a lot. Bye-bye.
Okay.
Thanks. Bye-bye.